Now we are going to talk about selection with QTL information in the breeding programs. So in the previous week we discussed about QTL analysis and association mapping, and basically those methodologies at the end, they are different, they use different approaches, but at the end they try to identify genomic regions that have association with the phenotype that we are interested on. And as you can see here, there are different kind of a phenotype or phenotypes that are controlled for different kind of QTLs. So we have some phenotypes that are controlled for major QTLs, and in those phenotypes basically we can see two different a group of phenotypes. Here, for example, we have one phenotype that have a lower and another one that have a higher phenotype of interest. So just say that that is disease resistant. So we have a lot of or one major QTL that control the resistance or susceptibility for a specific pathogen. Also, we can have phenotypes that have major QTLs, but as well have minor QTLs, and in those phenotypes, the difference between the lines that have the major QTL or the positive QTL and the negative QTL. When I say this, that negative or positive is the good allele and the bad allele, basically, for that QTL. And you can see here that the difference is not that evident like in the first scenario, because we have minor QTLs that control some of the variation in the data. So that is why we have a kind of a continuous distribution here. So there is not something that it's complete different phenotypes here, because we have a combination of several minor QTLs. And when the trait it's just control for minor QTLs, you can see here that we're going to have a continuous variation of the phenotype or a kind of a normal distribution. And in that kind of phenotypes, usually there are not major QTLs, or the major QTL for that trait is just fixed in the population. So that is why it's not there is no segregation for that QTL, major QTL in the population. So the variation that we can see in the phenotype is just due to the minor QTLs, and this it's like a representation of complex trait or quantitative trait. And now I want to talk a little bit about how we can use that information of the QTLs in the breeding program. So I just want to show you this data from the one paper of 2017, when they select one genotype that was resistant, so this is a biparental cross, and another one that is susceptible, and they made some crosses between those lines. And as you can see here, all the F1 line population that is heterozygous, it's resistant. So that it's evidence that the resistance for this pathogen that was anthracnose, it's controlled for one major QTR. And when they advanced another generation, they saw a segregation of a 3-to-1 ratio. So that means that again, that segregation in the F2 confirmed that the effect of this major QTR or this gene has a dominant action, and basically it's controlled just for one major QTR. And that kind of that it's the same example that we saw in the previous slide, when one major QTL control the phenotype. So when we have something like this in our trait, it's kind of a straightforward to identify the sources of QTL alleles. So in this case, as the traits control just for one QTL, we can easily identify one line that is susceptible and the another one that is resistant. Then we have to generate a population to try to again create some meiosis and recombination between the lines, and then we have to use molecular markers to try to identify those recombinations in the population. And when we have identified with molecular markers the QTL location, we have to confirm the effect of that major QTL in different backgrounds. And that it's really important because usually with QTL analysis, the studies finish like in the paper. So but not a lot of QTL analysis or the data of QTLs it's been used in breeding programs. So, and one of the reasons for because of that is that or one of the reasons of that is that the QTLs could be just important in the population that have been identified. And that is why it's important to check in different backgrounds, to check if a QTL, for example, that has been found in this population, also it's important in the other populations, because as I mentioned as well in the previous in last week, in biparental population, you are just testing the two alleles in diploid organism. So there are maybe other allelic variants in the other in other genetic backgrounds where the QTL could be not have the same effect, or could be not have an effect at all. So that is why it is important to check the effect of the QTL in different genetic backgrounds to assess basically if you can use that information in marker assisted selection to do early selections in your breeding program. And after confirm that a QTL, that QTL it's consistent across different genetic backgrounds, you can start to use that information. You can design some molecular markers in that QTL and basically try to use in early generations to select a germplasm in the breeding program, and try to increase that good allele in your population. But what happened that it's in the case that we have a major QTL. But what happens when there are many QTLs that control the trait? So when we have a lot of QTLs, we are going to have a continuous distribution of that phenotype, and that is what happened in a lot of traits of importance in plant breeding. For example, here I have some data of my research. I am working with mineral concentration, iron and zinc, and you can see here the distribution of those traits is quantitative uh, between years and locations. So, and we published a paper about the QTL that had been detected with association with mineral concentration, and we identified more than 100 QTLs. As you can see, there are a lot of QTLs, and like the heritability of the quantitative traits usually is not very high, usually is around 0.5, 0.4. So a lot of the environmental component it has a strong influence in the phenotype as well. So that is another reason why you can find some QTLs in some populations, but different locations for the same traits in other populations. So it's complex to work with this kind of traits. And that it's in the case of a mineral concentration that I just told you that we have identified more than 100 QTLs. But when we are talking about a yield that is I think more complex than mineral concentration, I'm working right now in a meta-analysis of the QTLs that have been identified with this trait that also, it's, as you can see here, it's quantitative, and I have identified more than 608 QTLs across the genome. A So that makes a really difficult the implementation of these or yeah, the implementation of the or the use these QTLs in breeding programs, because it's just a lot of information. and and to give you an idea about the power to detect QTL, that is another limitation when there are a lot of QTLs that control um, target trait. It's for example, here in this figure, if we have 10 QTLs, those are simulations. Here I just show you some simulations. So when there are 10 QTLs that control um, different phenotypes with different heritabilities, you can see that, and so here we have the population size, here we have the power, and in this first graphic, with only 10 QTLs, we have to increase until 1,000 individuals of the population, so have a population size of 1,000 to reach a power of 0.8. So usually a power of 0.8 it's good, lower than that is not that good. So as you can see here, with the heritability of 0.3, that is a common heritability for traits as is yield, for example, for complex traits. We have to have a huge population size to have enough power to detect 10 QTR, so a trait that is controlled for 10 QTR. But when we have more QTRs, so in this case we have 40 QTRs in this trait, you can see that when we have 1,000 individuals, the power is 0.2. And when we have like the heritability of 0.95, so that means that just 5% of the variation is due environmental factors. You can see here that with 1,000 individuals, we are going to have just a power to detect the 40 QTLs of 0.8. And that is just to show you the complex, how complex is identified all the QTLs. And in traits as yield, I think that it should be more than 100 regions or hundreds of regions that have association with that kind of a complex trait. So uh, that is going to be more difficult to detect. And here again, this is in a F2 population. So if you are using, for example, a diversity panel, the power is going to be lower and lower because the frequency of the alleles are going to be different, not as in F2 that hopefully you remember that is 0.5 for PM for Q. And like the take-home message here is that the power to detect the QTL depend on the population size and the heritability of the trait. So if you have a trait with low heritability, you should try to increase the population size. And if your heritability is really high, maybe your population size shouldn't be so big. But as you can see here, it's still with a really high heritability and high population or a big population size, it still is difficult to capture or to identify all the QTLs. And that also is important, this graphic here, because the issues are not just to identify all the QTLs, but also to identify the R square or the variant that are it's explained because the QTLs that you identified. So here again, with the same example of 40 and 10 QTLs, we can see that to identify the real R square, when we have 10 QTLs, we need a population size of 1,000, and it's the same for 40 QTLs. So when we are working with small populations, the R square it's a worse bias, has a worse bias to estimate the R squares. So in theory, or we are going to have an overestimation of the R squares for the QTLs that we can detect. And that is important. And another thing that I want to mention here is that notice that as more QTLs are involved in the traits that we're interested on, the effect, the R square, so the variance that is explained with each QTL is lower, and as well the uh, effect or the proportion that is explained for each QTL also, or the effect of each QTL in the phenotype is going to be small, as smaller if you compare a trait that is controlled for more QTLs that with less QTLs. So again, remember that we have these 4 steps to use the information that we can detect in a QTL analysis, or basically to implement QTL regions into the breeding program. So when we have a lot of or many QTLs that are involved, so the first step that is identified the sources of QTL alleles is not that easy because we saw that when we have one major QTL, it's pretty easy. We just select the lines that are resistant or susceptible in that example. But here, as there are hundreds possible, hundreds of regions that have association with the phenotypes, so all the genotypes are going to have good and bad alleles. So it's going to be difficult to identify the sources, just a source that have good alleles and another one that have bad alleles. So have contrasting parents, it's going to be challenging. The second point that is find the markers that are in LD with the QTL. Also, it's challenging when we have a lot of QTLs, because again, when a trait is controlled for several QTLs, the power to detect all the QTLs is gonna be small. So as you can see here, again with the same graphic, we can see that with 40 QTLs, if the trait is controlled for 40 QTLs, population size of 500, that actually it's kind of big. It's not able to detect all the QTLs when the heritability of the trait it's kind of low, and the heritability of quantitative traits tends to be between 0.3 and 0.5. So you can see here that the power to detect all the QTL is not so big. And then if we can identify some QTLs, we have to confirm the effect of that QTL into the different genetic backgrounds. So we're going to have a low power because first, the effect of the QTL in quantitative traits are not going to be so big. Then we're going to have interaction with the environment. So for example, if we are talking about a trait that have a heritability of 0.3, that means that 70% of the variation in that phenotype is because environmental factors. So that it's a really big issue to confirm the effect of one QTL in different genetic backgrounds. And then we have the QTL segregation. So if we have hundreds of QTLs in that trait and we just want to confirm the effect of one QTL, so that means that the other QTLs are going to have segregation in the population, and it's going to be too difficult to detect the effect of the QTL that we want to confirm. And then if we cannot confirm that, as you can imagine, we just cannot use methodologies as marker assisted selection to yeah, basically do selections using just markers. So the take-home message in this presentation is that major QTLs can be used in breeding programs as a you saw it is a common practice in several traits. So in common being, we use, for example, a marker assisted selection for anthracnose resistance, and it's an efficient way to select in early generations a germplasm that is resistant for this disease. But for quantitative traits, it's challenging because it's kind of difficult to identify all the QTLs that are involved in the trait that we are interested on. And then in case that we are able to identify those regions, it's going to be difficult to confirm the effect of those QTLs. And after that, if you think that maybe you are going to have 100 markers or more that have or are in LD with different QTLs, the implementation of a marker assisted selection with that number of markers maybe is going to be challenging as well for the plant breeding pipeline. So that is why predictive methods are maybe more useful in quantitative traits, and that is why in next video we're gonna start to talk about a genomic prediction. So this is the end of this video.